আচ্ছা, আমি শুরু করছি তাহলে। শুনতে পাচ্ছ আমাকে?
Yes, sir.
আচ্ছা, এটা দেখতে পাচ্ছ পোস্ট ট্রান্সক্রিপশনাল মডিফিকেশনস?
Yes, sir.
হ্যাঁ, post transcriptional modification. I'll start with this particular topic. So what is basically almost all major types of RNA synthesized by cellular DNA dependent RNA polymerase undergo changes before they carry out their functions. So all RNA which is formed from DNA dependent RNA polymerases, the RNA polymerase which transcribe on DNA, and all RNAs which are formed, they essentially এক সেকেন্ড এসছে। So they essentially when they are produced, there is some sort of modifications they occur. So the post transcriptional means after transcription, which modifications and processing which takes place. So these modifications actually involves addition to or alterations of existing bases or sugars. And this type of processing, it involves phosphodiester bond cleavage and loss of certain nucleotides from transcripts. So I will just show you what a phosphodiester bond cleavage is. If you look at this particular picture, this is a phosphodiester bond. This is the 3' OH and this is the 5' OH. And here OPO, this bond cleavage is known as, it's a phosphodiester bond. So when this bond is cleaved, this is known as cleavage of phosphodiester bond. এটা clear?
Yes, sir.
Okay. So this involves phosphodiester and loss of certain nucleotides from the transcripts. So once it's cleaved, so if you have a loss of nucleotides post phosphodiester cleavage. So basically, after cleavage, these little small nucleotides are formed. They involve phosphodiester cleavages. So if this is a phosphodiester bond, so you have 3 phosphates: alpha, beta, and gamma phosphates. So this when this particular thing DNA nucleotide takes place, Hmm. So, and there is a loss of certain nucleotides from the transcript. So the nascent RNA, also known as primary transcript, needs to be modified to become functional tRNA, RNAs, and messenger RNAs. And this type of modification is critical to eukaryotic system. So, what are the changes associated with primary transcript? So, mRNA in prokaryotes, modification, some people say there is polyadenylation, but no evidence so far, therefore question marks. There are some specific cleavages by endonucleases. The products are mRNAs itself. In eukaryotes, the heteronuclear mRNA or the primary transcript, it is 2 to 14 kb long in mammals, average size 8 to 10 kb, 4-5 times longer than a messenger RNA transcript. So what are the modifications which take place? 5' capping and methylation and 3' polyadenylation. And the processing is splicing out of introns. And the product is a functional RNA. Then you have the ribosomal RNA. In prokaryotes, there is a pre-RNA, methylation takes place and specific cleavage takes place. Result, you have the 16S subunit of ribosomal RNA, 23S and the 5S ribosomal RNA unit and spacer RNA, spacer tRNAs. In eukaryotes, it is a bit, the same thing happens. Only it is instead of 16S, 23S, it is 18S, 28S, and 5.S. 3 ribosomal RNA fragments are formed. In tRNA, there is a pre-tRNA. Many bases are special bases are formed. Many bases are modified. And there is specific cleavage by endonucleases, trimming by endonucleases, and there is a CCA end addition. Result is mature tRNA. Same. In eukaryotes, the same thing takes place. So we will go on to the modification of eukaryotic mRNA. What are there? So modification of hnRNA, that is the heteronuclear RNA. It is the heteronuclear RNA, heterogeneous nuclear RNA or hnRNA. They are larger than matured mRNA by many folds, around 4 to 5 times normal. They are larger than the mRNA. It's all coming out, goes out by splicing. So what are the modifications? The first modification is capping at the 5' end and tailing at the 3' end. Capping is 5' methyl guanosine cap and at the 3' end poly A tail addition. So what is this 5' end modification? Then 5' capping and methylation. There are two types of methylated caps. One is monomethylated caps, which are commonly found in eukaryotic RNAs, mRNAs, and you have the trimethylated caps, less frequently observed in some non-coding RNAs, but highly conserved throughout the eukaryotes. Let's talk about monomethylated cap. So the presence of an unusual methylated nucleotide at the 5' terminus, most viral and cellular mRNA, and it was discovered by A. Shatkin and co-workers in 1975. They found an unusually methylated nucleotide in most viral and cellular messenger RNAs in the 5' terminal. So the entire methylated terminal oligonucleotide is known as the cap structure. The site of biogenesis is in the nucleus by a series of enzymatic reactions take place. The capping process is completed before the completion of nascent transcript. So Before the nascent transcript is made by from transcription, the capping is already done. 5' cap of most messenger RNA is monomethylated. এই বালিশটা ফেলছিস কেন মামা? অসভ্যতা করছিস কেন? 5' cap of most mRNA is monomethylated, but some small non-coding RNAs are trimethylated. Monomethylated cap. So the primary transcripts, pre-mRNAs or heterogeneous nuclear RNAs, are usually first capped by a guanylyl group. And this particular guanylyl group, it is catalyzed by guanylyl transferase. Methyl guanosine residue is guanylyl residues. So capping G residues is methylated at the 7th position. Additional methylation occurs at the 2' O position of the next two residues and at 6 amino of the first adenine. Just hold on for a minute. বিছানাটাকে নোংরা করছো কেন? Okay. So this is a GTP molecule. This is the guanosine residue over here. This is the guanine residues, guanylyl residue, you can say, and it is attached to a sugar and triphosphate, 5' triphosphate. And this is the 5' end of the trans, some base, say adenine. And here it is the sugar and triphosphate. So this is the 5' end, absolutely the beginning. What happens over there is this guanylyl, this with the presence of this guanylyl transferase, this PP is eliminated out. 2 PPs are eliminated out and 1 PP remains. That gives the energy to this reaction, and basically it's another phosphodiester bond is formed. This is the 5' cap, and it happens at the 7th position. This is the 7th position of the guanylyl. Clear?
Yes, sir.
Yes. Okay. So if you look at the full structure of the 5' monomethylated cap, basically this is the cap O methylation, which is present everywhere. This is guanine and this is the cap over here. This is 7-methyl guanosine. This is the one which I've written in red. Okay, and this is the 5' end of the transcript. Here there is an OCH at the 2' OCH3. There is another cap. This is the trimethylated cap. You see that 3 cap. This is the cap 0, cap 1, cap 2. Gives more protection. It's very rare. TF2H kinase, it phosphorylates the 5th sulfur at the CTD tail and recruits enzymes required for 5' capping of messenger RNA. These are small questions which me ask. 5' capping enzymes include this particular enzyme, remember it. This is the primary transcript. Triple phosphate, nucleotide 1, nucleotide 2, nucleotide 3. RNA triphosphatase. PI is removed and PP. N1, N2, N3 is there. In mammals, this guanylyl transferase has 2 enzymatic activities. It removes the PPI. And then this 5 prime cap is attached over here. Then 7-methyl guanosine transferase takes a methyl group from SAM, S-adenosine methionine. It becomes homocysteine. The methyl group is given over here to guanosine at the 7th position of guanine. And then this cap was 0, cap 1, cap 2, again from another molecule is it's taken. And it is transported from the nucleus to the cytoplasm. The 5 prime cap structure is found on H and RNA 2. The capping process occurs in the nuclei. All the capping, it takes place in the nuclei. The cap structure of mRNA will be recognized by the cap binding protein required for translation. So there is a cap binding protein during translation in the ribosome, and the capping occurs prior to splicing. Before splicing takes place, this capping occurs. These are the small questions they will ask. Yeast mRNA have cap 0. Slime molds have cap 0, but 25% have cap 1 structure. That is the first nucleotide at 2' position. There is another methyl OCH3 group. The messages or mRNA of brine shrimp and sea urchin, they terminate with cap 1. Mammals have high percentage of cap 1 and cap 2. Between the cap and translation initiation codon, AUG, there is a length of non-translated RNA known as the leader sequence, UTR. The length of the leader sequence varies from mRNA to mRNA. It varies. In nucleus, cap is recognized by cap binding protein CBP20/80 heterodimer, which stimulates splicing of first intron and also interact with TREX complex to facilitate mRNA export out of the nucleus. In the cytoplasm, the cap interact with elongation initiation factor 4F to initiate translation process. You have to remember all these points. This is in translation, you have read elongation factor 4. So the cap, it before translation, it interacts with this elongation initiation factor 4. Ribosomal RNA and tRNA are not capped. So what is the function of the 5' cap structure? The cap may function in the transport of mRNA from nucleus to cytoplasm. The CBP20/80, it interacts with TREX components and facilitates the transport of messenger RNA. It protects mRNA from phosphatases and nuclease attacks. So basically, it protects different exonucleases attack, which will digest the mRNA. Rather, it contributes to the stability of the messenger RNA. And it is required for the productive mode of elongation of transcription by RNA pol 2. It helps in ribosomal attachment. It may play a role in splicing. The leader sequence and cap binding protein CBP may play a role in enhancing and stabilizing the interaction of the mRNA with the ribosome and translation initiation process. It is required for efficient translation. So this we will do in the next day's class. That is tomorrow. Okay. তোমরা বুঝতে পেরেছো?
হ্যাঁ স্যার।
ঠিক আছে, পরের দিন আমরা ওই বাকিটা নিয়ে শুরু করব। ঠিক আছে, আজকে রাখছি।
থ্যাংক ইউ স্যার। থ্যাংক ইউ স্যার।
ইউ ওয়েলকাম।